During the initial steps of mineralization, cells produce extracellular matrix proteins and release matrix vesicles that accumulate calcium and phosphate to facilitate apatite nucleation. In this report, we compare the nucleation of minerals by two selected human cell models, osteoblastic hFOB 1.19 and osteosarcoma Saos-2 cells. Their mineralization profiles were analyzed by different methods. Put all materials under the lamina and sterilize them under UV light. Change the medium to 10 ml of fresh culture medium with fetal bovine serum. Resting cells are incubated without stimulators, while stimulated cells are treated with both ascorbic acid and beta-glycerophosphate. First, gently drop ascorbic acid to final concentration of 50 micrograms per milliliter onto the surface of the culture medium to stimulate the cells. Next, add beta-glycerophosphate to final concentration of 7.5 millimolar in culture medium to stimulate the cells. Gently stir the culture dish and put it into the incubator for 7 days. After 7 days of incubation, wash the cell cultures with phosphate buffer saline, PBS. Add 5 ml of 2% Alizarin Red and incubate the plates for 30 minutes to stain the minerals. Wash 3 times with PBS. Carefully add PBS to the dish wall. Try not to destroy the minerals. Observe the minerals under an inverted light microscope.